Hello, everyone. I'm happy to be here today at BBCon 2024. My name is Matteo Tardelli, and I'm a senior biomarker scientist at Owlstone Medical. Today, I'm going to talk about our study called Analyzing Postprandial Response Using VOCs in Exhaled Breath, a longitudinal study slide. Just a bit to the project overview here. We asked our volunteers to fast overnight and come on site to give multiple samples before, in a fasted state, and after, in a fed state, a liquid meal. The liquid meal was defined as a orange juice and a UA protein drink. In this longitudinal study, we actually aim to elucidate first the impact of dietary intake on exhaled breath VOCs, also highlighting the capability of our OmniBreath collection and analysis platform to detect metabolically relevant volatile compounds. We also wanted to non-invasively detect diet-induced metabolic changes in breath to really facilitate disease-related study in the field. We processed the samples on our platform, which is a TDGCMS platform, and did feature extraction on untargeted data. Just the study population and protocol here, we defined some inclusion and exclusion criteria. The inclusion criteria were the patient had to be able to provide written consent for, they had to be an adult of age between 18 and 65. The BMI had to be between 18 and 30. They had to be able to fast for at least 8 hours before sample collection, and they had to not brush their teeth on the day before collection itself. They had to be able to provide a finger stick for glucose measurements and willing to be able to consume a provided meal between breath samples as well. When it came to the exclusion criteria, we actually wanted to exclude current smokers. We had to exclude participants that clinical procedures or surgeries within 10 days prior to sampling. And we also wanted to exclude volunteers with significant comorbidities, such as type 1, type 2 diabetes, as well as some inflammatory diseases, such as IBS and IBD. You can see on this table on the right the different phases of our study. In the screening phase, we assessed for eligibility and took height and weight, did some demographics questionnaires, as well as recorded medical history. Once they passed this part, a finger stick glucose was taken in the pre-sampling phase, and volunteers they were asked to rinse their mouth with water. Then we started with the real sampling phase, in which we asked our volunteers to take 2 samples in a fasted state. We then asked to take a meal that had to be consumed in 20 minutes, and then we took another 2 samples, what we called fed samples, at 20 minutes and 1 hour after the first meal. We did the same on day 2 and day 3, just without the meal part here. And I think you can appreciate this design better in this scheme. And I wanted to discuss quickly on the numbers we got here. Especially on day 1, we got 20 volunteers that we were able to enroll. As you can see from the table here, the distribution of the population was pretty balanced in the group. And also in the pie chart here, you can see that we had 60% of females and 40% of males. Their BMI was actually comparable, as well as their average age, and the blood glucose was pretty comparable across the groups as well. Just as a brief statistical overview, we actually used different tests for different purposes. We used 3 metrics for determining on-breath VOCs to really distinguish what's a background noise versus a genuine biological relevant breath VOC in our population. These 3 metrics were the standard deviation method, a parity test method, and a ROC AUC method. We also used the Friedman test and post-hoc testing Wilcoxon signed rank to really compare the single biomarkers between groups on day 1. We did some clustering analysis, and we also did some Spearmonk's rank, which served for identifying a correlation between glucose and fasted state. To the results, we actually in our untargeted feature extraction, we found a final data set of 8,157 molecular feature, of which 687 passed the sparsity criteria. 217 were actually identified on breath, And you can see on the Venn diagram here on the right how many VOCs actually were on bread by each metric. So these will be the 50% plus mean plus the 3 standard deviation metric, the pair t-test metric, the ROC AUC metrics, and in the middle, this will be like all of those together. To get to the biology interpretation of this study, we actually saw, as expected, a large increase in several flavor and aroma compounds coming from the fasted to the fed state. You can see the table here on the right describing those IDs and mesh compounds, the directional change, the group comparison, as well as the effect size and p-value here. Possibly as expected, we saw a large increase of limonene and valencine already at 20 minutes, which are known to be the most abundant VOC in orange juice. You can see here in the bar plot at the bottom this huge increase already at 20 minutes that we can see in the postprandial phase. We also observed increase in other monoterpenes such as alpha-pinene and beta-pinene, as you can see here in the bar plots, as well as cinnamon flavor compounds that were part of the protein juice. From an exposure perspective, these compounds were actually found to be increased in the postprandial phase. These were furans, 2-pentylfurans, as well as 4-dimethylfuran. We think that these furans were actually formed during thermal processing of food containing sugar or ascorbic acid. So they could really be a part of the pasteurization kind of methods of which these two drinks actually went through. So these were also interesting compounds that we found out in the exposure-related kind of buckets. Of interest, we found other microbiome-related compounds in their targeted feature. We found quite a bit of short-chain fatty acids, such as the butyric acid and propionic acid and acetic acid, which showed an upward trend in the postprandial phase already at 20 minutes, as you can see from the bar plots here at the bottom. This was somewhat surprising, but aligned with an internal study that was run at SIFT-MS that observed a spike in ethanol and propionic acids already at 15 minutes after the administration of water and glucose. So we really think that short-chain fatty acid, it may arise from simple carbohydrate hydrolysis or amino acid metabolism by the oral microbiome. And this will take place already at 20 minutes and 1 hour time points. Other microbiome related that we found to be interesting in this data set were 1-propanol and 2,3-butadiene, which also showed an increased trend. Instead, indole and 3-methyl-indole, which are actually products of the fermentation of tryptophan, were actually decreased or remained unchanged after food intake. This was actually in keeping with a study that observed a negative correlation between exhaled indole and blood glucose when type 1 and type 2 diabetes were actually sampled for 6 hours after standardized meal. Other compounds that we found in the untargeted analysis were octane. Octane is an inflammation-related VOC, which is actually produced endogenously by the lipid peroxidation in cells, which is driven by oxidative stress. From a biology perspective, any food can actually cause oxidative stress in a postprandial phase. So any high fat or high carbohydrate meal can actually drive that. So this was somewhat expecting and reassuring that our breath VOCs were actually coming from a biological, meaning an endogenous phase. We also found some decreases in sulfur compounds, namely dimethyl disulfide and dimethyl trisulfide, which are actually associated with bad or morning breath. So these went down in the postprandial phase. Other compounds were going up, and the origin of this compound is actually unclear at the moment, but we'll do further digging into that to understand if they're microbiome related or exposure related compounds. We tried to connect the dots between VOC and amino acid metabolism, as we knew that the UL drink had quite a bit of amino acids in there. We connected metabolic pathways such as tryptophan degradation and metabolism by bacteria and the production of indole and methyl-indole. Other amino acids such as valine, isoleucine, and leucine actually undergo catabolism and produce metabolites such as propionyl-CoA. This is a TCA cycle intermediate and also a precursor of short-chain fatty acids. And this itself can be converted into propionic acid and other short-chain fatty acids. Also, 1-propanol actually could be linked to the metabolism of threonine, which is an amino acid that is part of the urine drink. And this can be converted itself in propionil-CoA and then subsequently in propanol. We decided to run an enrichment analysis on metaboanalyst and with the compounds that, of course, were found to be significantly upregulated in our dataset, and found out that the phenylalanine metabolism pathway were actually highly enriched in our dataset. We know that phenylalanine is highly present in the UL drink, and it is metabolized into various compounds, including phenyl acetate and phenylacetaldehyde, both of which were actually elevated in our dataset after feeding. We also did an association of VOCs with fasting glucose, and we found out that 11 VOC in our dataset significantly correlated with fasting glucose. 7 of them were inversely correlated, while 4 of them were positively correlated. Of mention, 2-butanol were actually positively correlated to fasting glucose, as you can see by this graph here on the right. 2-butanol is actually an aldehyde that can be produced during lipid peroxidation. And we think that the higher glucose levels may increase oxidative stress, leading to more lipid peroxidation. So it will make sense in our eyes to see this positive correlation between the fasting glucose itself and 2-butanol. There's another example that's instead 3-methyl-indole or scatol, which showed a negative correlation between the VOC itself that we measured in breath and the fasting glucose. This was somewhat consistent with previous study that found that fecal scatol levels to be inversely associated with fasting glucose, and the study in breath in type 1 and type 2 diabetes that we actually described before in the previous slide. We also saw 2-butanol and MEC to correlate with fasting glucose. We know that MEC is generated by isoleucine catabolism and also metabolism of 2-butanol, which is present at low levels in the body. As you can see from this scheme, we really think that increased glucose level may favor the minor regulation pathway of MEC to butanol. So to summarize the findings today, we saw a significantly increase of terpenes like limonene and valencine, along with other monoterpenes after food intake, indicating a possibly expected exposure-related response to the food intake. Changes in volatile sulfur compounds, as well as some products of amino acid metabolism and microbiome-related metabolites, including short-chain fatty acids, possibly indicate a diverse metabolic and microbial activity post-ingestion. We really think that these microbial products possibly will come from the oral microbiome rather than the intestinal microbiome. We think that there was possibly not enough time for the meal to reach the gut and get some fermentation going on that was genuine to see in breath. We also saw that 11 VOCs were actually associated with fasting glucose levels, including 3-methyl-indole, replicating the findings of a previous metabolomic study as well. And with this, I thank you, the audience, for the attention, and I'm happy to take any questions you might have. Thank you.
Excellent. Thank you so much, Matteo. So we've got a few questions that have come through. So first one is, did you examine the influence of lifestyle factors such as physical exercise on breath VOCs in this study?
Yeah, so that's a good question. So we definitely recorded some lifestyle factors here when the volunteers came in. We asked them to tell us their recent meals as well as what they drank before, quite in detail. So we really wanted to know and align on the kind of like drinks and meals they had before actually assembling with us as well. The physical exercise, I think it's an interesting question. I think there's a lot of interest towards that. I mean, we also recently published this ultramarathon paper that was really interesting in that direction. And so we also within the postprandial phase, we see quite a lot of oxidative stress kind of markers going up. And we're really wondering if there's anything like physical activity that could be driving that apart from the meal intervention itself. So I think at this point we're really thinking of possibly going back to the volunteers, asking the question whether they were active at the time of sampling, and really try to understand if there's a way to stratify the fasting population and then really see if there's any kind of impact on the on some oxidative stress markers also in these regards. So also really focusing on the physical activity side of things as well, rather than, you know, the postprandial state in this case as well.
Excellent, thank you. Next question is, did you identify any VOCs that correlate with BMI or are relevant to metabolic diseases?
So the BMI, if you remember for our scheme before, the BMI was actually pretty balanced across the groups. So we really didn't see a lot of difference there, because we just on purpose kind of you know, excluded patients that BMI above 30 as well. So we didn't really see like interesting correlations in that regards. I think in regarding the interesting microbiome metabolic relevant VOCs, we surely see you know, a lot of short chain fatty acid and indoles and other microbial products there, which of course are really relevant in many metabolic disease states, you know, starting from type 2 diabetes going into IBD and IBS as well. So I think from that aspect, there were quite a bit of exciting kind of VOCs we could see in breath in the postprandial state. So yeah, surely like worth a deeper dive on that one.
Excellent. Next question is, do you have measurements of glucose levels in the postprandial state that could be correlated with the VOC data?
Actually, we didn't do that. We just did the fasting glucose measurement in the fasted state, and that's what we did correlations on. So all the correlations and the lines that you see possibly in the last slides as well were actually in regards to the you know, the correlation to the fasted states and the VOCs that we saw in breath. So yeah, unfortunately, we didn't really take the you know, the glucose stick after the lunch. However, we would expect also by seeing the fasting glucose that volunteers were actually healthy. So possibly there was not going to be such a big difference as we saw in the fasted state as well.
Excellent. And following up on that, so I was interested to see that previously reported result from the team at Roche of indole decreases, or sorry, yeah, indole decreases as the blood glucose increases. I was wondering if you could expand a little bit further on the mechanism there. And, you know, do you see measurement of that type of compound as being potentially useful as a way to try and indirectly measure blood glucose levels, as opposed to, you know, having to do finger pricks or continuous monitoring?
Yeah, it looks like a possible avenue as well in terms of non-invasive kind of you know, measurements of in a way that you can then infer like the glucose levels in blood as well. In that paper from the Roche group, they actually did a very good job within that. So they were really looking into these type 1 and type 2 cohorts and really really trying to figure out whether, you know, indole could really serve there as a proxy possibly for blood glucose as well. What we see in our data sets is pretty similar actually. We really see a decrease of indole and as well as cattle, so the 3-methyl indole as well, really going down dramatically after food intake. So we really think that, yeah, possibly that's you know, a way that with our results we could really reproduce that piece of literature as well. So yeah, I guess you know, future kind of investigation within that space could really be interesting to really assess whether, you know, kind of breath indole could really be a proxy for blood glucose, but just in a non-invasive manner as well.
Excellent, thank you. One point I'll make just before moving on to the next question. One of the things, obviously, you have a big advantage of breath is you can pick when you collect the breath sample. So you mentioned, you know, earlier time points, you more likely have contribution from the oral microbiome. Then over time, you know, as substrates transit through the GI tract, you effectively have the ability to probe different, you know, different microbiome compartments essentially. So I think that's a huge advantage of breath relative to, you know, a fecal-based sample where you might just look at the presence of the bug but not the activity at a particular point in time. So the next question here that kind of relates to that is, to what extent do microbiome-related metabolites in the breath reflect microbial metabolism in the small intestine and the large intestine, respectively? With intake of foods, this would result in movement of food in the colon as well, changing absorption, I presume.
Mm-hmm. That's a super interesting question. We've been actually asking ourselves quite a bit about that as well. I think, according to our time points, that were really like 20 minutes and 1 hour after the meal, it is possible that, although the meal was, of course, liquids, it is possible that there wasn't not enough time for the microbiome to really interact and kind of ferment, possibly just digest the carbohydrates and amino acids present in that meal, but not really ferment these products from an intestinal perspective, of course. So I think, yeah, it would be really interesting to understand whether, just by modifying the time points, like possibly allowing a little bit longer, like waiting a little bit longer, possibly up to 6 hours as well, if we were able to see the same signal, or if the signal within this kind of microbiome interesting compounds will be lower or higher of what we see right now. Of course, like one approach could really be that to really make sure that we exclude the oral microbiome from the kind of the discussion here. It will really be that after the volunteer actually drinks the meal or whatever intervention we're thinking of, getting them to possibly brush their teeth or remove like this first biofilm that it will create with the meal intake itself, and then possibly like again focus on like longer time points to really make sure that we get a genuine kind of intestinal signal there as well.
Yeah, there's a question related to that, and how long before or after meal would be best to obtain a breath sample? I suppose I'd say it depends what you want to check. If it's oral microbiome versus small intestine versus colon.
Absolutely.
One question here is presence of chlorinated organic compounds is interesting. Any ideas where these are coming from?
Not really. No, I don't. No, I don't think that I have an answer to that one. Yeah, I was just wondering. Yeah, which one they meant with the chlorinated compounds. Might need to go back to the slide and check it out. But no, I don't really right now know that. Yeah.
Okay, no worries. So next question, were you able to determine the length of fasting needed for such studies?
So we actually tried to standardize that quite nicely, across the volunteers as well. We asked at least 8 hours of fasting before actually the sampling to take place as well. But yeah, essentially people, from what I recall from the metadata as well, people would kind of have dinner at around 8 p.m. and will come basically to the office, to the sampling facility around 9 a.m. So it will be like more likely around, like 10 to 12 hours kind of fasting before the sampling to take place as well. And I think this is really important when you're then considering also design of, other trials and clinical trials in the breath space to really like try to get that standardized, because as you saw, like there were a lot of like exposure related compounds like really shooting up after the meal intake. So that's something really important to figure out when you're planning a clinical trials in breath as well.
Yeah, it's interesting. Obviously, some of those compounds you introduce are things that are useful in other contexts as well. So our friend quinine, which is metabolized in the liver, as we'll kind of see in other studies throughout the next couple of days. So the next question here, it might be a naive question, but in slide 14, how true is it to discuss the increase in or decrease in correlation trend? The data looks more scattered.
Yeah, I think we actually run all the tests needed for that correlations, and I don't know if you maybe noticed there was also like a correlation like numbers there and things like that. So yeah, surely like if you have the chance to review that, I'm sure if you figure out that there was significant the correlation between glucose and the VOCs.
Yeah, all the slides and talks are available afterwards as well. So if anyone does have follow-up questions on that or anything, do just let us know. Last question, actually probably time for two. So this kind of leads on, I think, from the previous talk. So would it be possible or recommended to include inborn metabolic disorders or inborn errors of metabolism in future in this type of study?
I mean, there's a lot of compounds that could really be interesting in that space. I think the main challenge here is really kind of sample, infants and newborns with the device, right? So there's some challenges really coming with the, kind of the size of the mouthpiece that you use and things like that. But possibly it's a very interesting kind of avenue to pursue as well. Like, yeah, I think the only challenge is really like coming down to the, I guess, device itself and the way that, the biomarker that you want to identify for this inborn metabolism is kind of solid enough for that specific disease you mean there. So yeah, a couple of challenges there for sure, but surely an interesting point.
Excellent. So I think we'll wrap it up there. We've answered pretty much all of the questions. So a big thank you, Matteo, for presenting the work today.
Thank you.
We'll make sure everything is available for everyone afterwards.